Pharma Ventures, experts in deals and alliances.
Hello and welcome to Pharma Ventures Insights here in London. Immuno-oncology is the key area for new drugs in treating cancer. Jim Allison was one of the founding fathers of immuno-oncology and checkpoint inhibitors, and he likened it to removing the brakes from the immune system. But we all know if you take the brakes off a vehicle, it could end in disaster. So there are many flavours now emerging of immuno-oncology treatments. And one company focused on some different approaches to this is Cioxus. Joining me today on the show is John Beadle, CEO of Cioxus. John, welcome.
Good morning, Adrian.
So John, Cioxus, an Oxford-based company, you've been around a little while, but perhaps you could tell us a little bit about the company and what it's doing.
Yeah, so we've focused on what's called oncolytic viruses, viruses that specifically replicate within tumour cells, but not within normal cells. And as you say, we've been around for a while. We've been working on a virus that is catchily called N-adenotussirev. It's a chimeric adenovirus that we've been able to show in the clinic we can deliver intravenously. It goes to tumour cells and replicates only in tumour cells. So that virus we have in the clinic in collaboration with Bristol Myers Squibb, looking at combination with checkpoint inhibitors. But importantly, because we've got a virus that only replicates within tumour cells, we believe we've got a gene vector system for delivery of genes of interest to tumour cells. And that really opens up the whole possibility of gene therapy for cancer.
So we'll come back to the gene therapy because that sounds quite exciting. So you have a deal on the oncolytic viruses. That sounds like a different virus to those that others have been using. Is that the case?
We have two different deals with Bristol Myers Squibb. So the first is a clinical trial collaboration with what we call an unarmed virus. So there are no genes in that virus other than viral genes. And then the second deal that we have with them is part of this gene therapy for cancer concept, where there are two co-stimulatory genes that the virus then delivers to the tumour, forces the tumour cell to express those proteins on the tumour surface, which then activate T cells. So it's an immuno-oncology approach, but a very different approach to, for example, the checkpoint inhibitors.
So it's priming the tumours to be recognised by the immune system in a way?
Essentially, it turns T cells on in an antigen agnostic way. So actually what we're doing is the mirror image of the CAR T cells. In the case of this particular virus, which is NG348, this particular virus, it's the mirror image of CAR T cells. So with a CAR T, you take the T cells and you genetically modify them with viruses and then you reinfuse them. With our approach, you're actually genetically modifying the tumour cell to interact with T cells. But in our case, we do it in situ, so this is an off-the-shelf product, avoids all of the autologous processing of cells, the manufacturing issues, delivers to solid tumours, and then activates the T cells. But the other thing that's different to the CAR T approach is that it's antigen agnostic, so we don't need to select out a CD19 or a New York ESO1 antigen. It will activate all T cells within the tumour.
And what makes this specific for tumours? Because that's also been an issue which people have had to get around.
Yeah, so this particular virus will be delivered to a range of different cells, but it only replicates within tumour cells. So it's replication deficient in non-tumour cells. But the genes of interest are driven off of the replication cycle. So you only get those genes expressed when the virus replicates, so it becomes tumour-specific gene delivery.
Right, and did that get you across any regulatory concerns that you were delivering viruses to healthy cells, effectively?
Well, I think that's the thing that you always have to be concerned with. In our case, the toxicology package that we put together is we take a virtual human, we take as many different human cells as we can, and show that we don't get these genes delivered to normal cells. So that's part of the regulatory package that we've put together both for Europe and for the US.
So another key difference, I guess, from CAR T is CAR T is almost exclusively focused on liquid tumours, but you're going after solid tumours.
That's correct. So obviously CAR Ts, second, third, fourth generation CAR Ts are coming through. and the ability to get the T cells into the tumour and to get the activity within a solid tumour is a key barrier. We are going after solid tumours. Our virus actually doesn't replicate in most hematological malignancies. It replicates within solid tumours, and we've shown delivery of the virus to solid tumours. So that's the reason that we're focused there.
Right, and that highlights another differentiator, and a positive one for you, I think, which is there are some other companies doing a similar thing to you, but they have to inject direct into the tumour. You're an IV.
That's correct.
Approach.
So most viruses don't survive in the bloodstream. So it's obviously possible to inject many tumours with a needle and syringe, but many patients have metastatic disease, lesions throughout the body. Intravenous delivery, systemic delivery is the best way to deliver if you can. And we've been able to show clinically that this virus does survive in the bloodstream and does go to tumours throughout the body.
And you mentioned you're doing a trial with a checkpoint inhibitor. Are you dependent on the checkpoint inhibitor to make this work, or is it just beneficial to have it?
So it depends on how the virus is armed. Some of the arming approaches might particularly suit a checkpoint inhibitor. So for example, you might arm a virus to bring T cells into the tumour. You might activate those T cells, as you said in the introduction, putting the accelerator on for the car at the same time that the checkpoint inhibitor is taking the brake off. In other cases, it may even be possible to deliver a checkpoint inhibitor in the virus. That's something that we've done. Not in any of our clinical candidates, but in preclinical candidates, we can put an anti-CTLA-4 or an anti-PD-L1 antibody or antibody fragment into the virus, actually force the tumour to produce the antibodies.
This sounds like a very elegant approach, John. And you mentioned the gene therapy angle as well, which is your platform, I believe. And like maybe oncolytic viruses, and people tried that in the past and failed. And then checkpoint inhibitors came along, opened a new door and a new approach to this. Again, do you need checkpoint inhibitors for this, or again, is this just a completely independent approach?
It could be independent, it could stand alone. We strongly believe that the approach that we've partnered with BMS, the 348 virus, could well stand alone independently of checkpoint inhibitors. But I think checkpoint inhibitors have created for immuno-oncology a fairly standard approach that other approaches will build on. So I think we'll see monotherapy approaches, but we'll also see combination therapy. I think combination therapy in immuno-oncology is probably here to stay. It's how we build that out with different approaches.
And there's a desire to have more personalised, foundation medicine driven approaches to cancer and treating the individual patient, which CAR-T does, and it's autologous, and we know that causes some issues. You're not taking that approach. You're taking, this is one size fits all.
That is true. So our approach, the virus will replicate in most solid tumours. We've been able to show that preclinically. We're obviously in the process of showing that clinically. So from that perspective, it's not personalised. But it is personalised from the point of view that an oncolytic virus bursts the patient's tumour cells in an inflammatory way. And that sort of sets up, we believe, a cancer vaccine in situ. So you expose those tumour antigens within the patient. In that case, it's personalised, it's the patient's own tumour antigens, but it's happening in situ. So our product is off the shelf, but the personalised anti-vaccine approach is within the patient.
So let's talk a little bit about the company a bit more. You've done a few deals, and did that one-size-fits-all approach, I called it, did that mean you got a more favourable reception from Big Pharma because they like that more than personalised approaches?
I think it certainly is a model that fits Big Pharma better than the personalised approaches, particularly when you've got a large component of cell manipulation in terms of manufacturing, for example, the CAR-Ts. So I think that's certainly true. There are certainly some players, as we know, Novartis, that have moved into that area, but others that are much more reticent. And I think the off-the-shelf, one-size-fits-all approach does fit Big Pharma. I think the flexibility of our platform and what can be done with it is another thing that really attracts them.
And I mean, no deal is easy to do, but was this a relatively easy deal to do then? Or,
as you say, no deal is ever easy to do. But we're working exceptionally well with Bristol Myers Squibb. They're a great partner to work with. We're looking to expand in terms of the deals that we do. We're looking to do other deals, but certainly BMS have been an excellent partner for us.
And that's set you up nicely for the future. So let's talk a little bit about the future and where the company is going and wants to go.
So we're taking a dual track from this point of view. We want to take our own products forward into the clinic. We're looking at taking at least 2 more programs into the clinic in the next year, with a third to come through in the year after that. So 2 of those we would look to be our own products, and a third we'd look to partner with another pharma partner.
And is this being funded by the revenues you're generating from the deals you're doing already, or have you got more money coming in from elsewhere?
We still have the investment from our last round, but obviously the upfronts that we received on the deal, or the deals with BMS, have obviously extended that runway significantly. 2018 will be another year of fundraising for us. We will add to that pot. And we believe we'll do that through further partnerships, but also through an equity raise.
Right, so you're not looking at an IPO right now?
Not in the immediate future, but it is something that we're starting to look at on the horizon.
So we should expect Cioxus to grow, become big, become a mainstream player in immuno-oncology, or is it going to get swallowed at some point by one of the guys? Everybody has their price.
I'm not going to predict the future, but our intention is to stay independent and to grow the business, to be a leading immuno-oncology player. We believe we've got a platform technology and a pipeline of products that certainly allows us to be a significant player in that space.
So where Immunocore have blazed a trail, you're going to follow on behind and do even better.
I think now is the time for UK immuno-oncology companies to go that little bit further than UK companies have previously,
and actually reap the benefits of the great science that occurs
Absolutely.
in the UK.
I'm a great fan of the UK science space. I think we produce great science. I think we need to do a bit more in terms of commercializing that, taking that further, being global players rather than just UK players.
Well, John, we wish you every success with your trials and in the future and in your developments of your company. Thank you for joining us.
Thank you very much.
For more information about Pharma Ventures, visit our website